Extraction. Before DNA profiling can be done, it must be extracted from the intracellular material. Routine forensic sampling from which a DNA can be extracted as are blood, body tissue, bone marrow, hair root, saliva, semen, skin, sweat, tooth pulp. It may also be extracted from biological materials deposited on various substances including chewing gum, cigarette, clothing containers, drinking straws, envelope flaps, face mask, gloves, and postage stamps. Also from a personal items such as combs, hair brushes, razors, toothbrushes. Extraction may be normal or differential. Normal extraction technique is employed when there is no intermixing of samples. DNA is extracted in the following step: cell disruption, cell lysis, breaking open the cell, exposing the DNA. This is done by alkali treatment, boiling, enzymatic digestion with the proteinase K. Grinding materials such as bone and teeth are frozen in liquid nitrogen and then ground to a fine paper with a mortar. Sonication. Application of ultrasound to disrupt cell membrane. Removing nuclear and mitochondrial membrane done to release DNA. Methods used are adding detergent, sarcosyl and sodium dodecyl sulfate. Sodium dodecyl sulfate. This dissolve membrane lipid, denaturate the protein and dissociate protein from DNA. Separation of DNA from a proteins and cytoplasmic contaminant. Lipid methods are adding high salt concentration mixtures dissociates nuclear protein as a histones from DNA. Using phenol chloroform isoamyl alcohol mixture in a ratio of 25:24:1. What is that? Phenol chloroform isoamyl alcohol. Phenol chloroform isoamyl alcohol. Okay. In a mixture of 24:1. Phenol chloroform form the organic phase in which lipids are dissolved. Isoamyl alcohol reduces forming. DNA dissolves in aqueous phase. Proteins are concentrated at the junction of the two phases. So in between, proteins will form. DNA dissolves in aqueous phase. Using solid silica surface, DNA is reversibly adapted to silica in the presence of a high concentration of chaotic salt denaturing agents such as guanidinium salt, guanidinium thiocyanate, and guanidinium hydrochloride. Silica is then washed with ethanol, which removes the chaotropic agent as well as cellular contaminant. DNA is then eluted, separated by rehydration with aqueous low salt solution. Eluted DNA is a double-stranded. Next is a preservation of DNA from degradation. This is done by adding a buffer system such as tris hydrochloride maintains pH in a range that prevents activities of a degrading enzyme. Reducing agent example mercaptoethanol, dithiotritol, DTT, to inhibits the oxidation process that can damage the DNA. Chelating agents example EDTA or Chelex capture divalent metal ion magnesium, which are cofactors of endogenous DNases that catalyzes the hydrolysis of DNA. Precipitating the DNA with an alcohol using ice-cold ethanol or isopropanol is employed. Since the DNA is insoluble in this alcohol, it will aggregate together, giving a pellet upon centrifugation gives a typical DNA amount recovered from the common sample. Compare figure with the fact that typical DNA required for a restriction fragmental length polymorphism is 50 to 100 milligram for STR, 1 to 10 milligram with PCR amplification. We can get this amount from just 200 microgram for reverse dot blot, 2 nanogram for YSTR, 0.1 nanogram. Each haploid cell spermatozoa contains 3 microgram of DNA. Each diploid cell contains 6 microgram of DNA. About 333 spermatozoa or 167 buccal cells would contain 1 nanogram of DNA. Thank you.